Hello and welcome to another episode of Bio Lessons to Go. I'm Mr. Dove, and today we'll be looking at some alternative forms of photosynthesis. Now, plants are a very diverse group of organisms, and they're able to live in all sorts of various environmental conditions. One reason they're able to do this is that multiple modes of photosynthesis have evolved. Now, the traditional photosynthetic pathway is called the C3 photosynthesis. And this is because when rubisco combines Ruby P with carbon dioxide during Calvin's cycle, one of the first compounds to be produced is a 3-carbon substance. Some examples of some common C3 plants are wheat, rice, and oats. Now, C3 plants have a very distinct leaf anatomy. The middle of the leaf, the mesophyll, forms distinctive layers. In this case, the top layer is called the palisades mesophyll, and the lower portion is the spongy mesophyll. These layers surround our vein, which contains bundle sheath fibers surrounding that vein that lack chloroplasts, and so they're unable to do photosynthesis. All of the photosynthesis happens in the mesophyll cells. Now, on hot or dry days, the stoma of the leaf needs to close in order to conserve water. As a result, there's going to be no new carbon dioxide going in, so there's going to be very low carbon dioxide in the leaf. Additionally, without the stoma being open to release oxygen, oxygen is going to be building up inside of the leaf, which once again is going to cause some problems with photosynthesis. Normally, when carbon dioxide concentrations are high enough, rubisco is going to be able to help fix carbon dioxide and mix it with Ruby P, which is present in the stroma, to produce our initial 3-carbon substance, PGA. Then, using the energy of ATP and the high-energy electrons that are coming from the light reaction, we're able then to produce that very usable sugar of PGAL. That usable sugar can then be used to produce glucose and other carbohydrate compounds for the plant, and then what's left over can be recycled to produce more Ruby P. Now, unfortunately, when the stoma closes, our carbon dioxide levels fall and our oxygen levels increase. And under these conditions, a process called photorespiration is going to occur. During photorespiration, rubisco is actually going to combine with the high oxygen. Rubisco has both an affinity for oxygen and carbon dioxide, much like our very own hemoglobin. Here, rubisco with the oxygen is going to produce a 3-carbon and a 2-carbon substance. The 2-carbon substance is going to leave the chloroplast and go to the mitochondria, where sugar is going to be used to produce carbon dioxide. It's going to be broken down to produce CO2, but unfortunately we're not going to produce any ATP in the process. So not only does photorespiration not produce sugar, it actually breaks it down. And even though we're going through the respiration process, we're not making any ATP, so it's a very inefficient process. So as a result, to limit photorespiration, what we found is alternative pathways of photosynthesis have evolved over time. One such example is C4 photosynthesis. C4 plants are able to do both have a C4 and a C3 pathway. In order to accommodate this, their leaf anatomy is a little bit different than our traditional C3 plants. In the mesophyll cells are arranged in rings around our bundle sheath that surrounds our vein. And the bundle sheath itself actually has chloroplast that's able to do photosynthesis. So we're going to have photosynthesis happening partly in the mesophyll and part in the bundle sheath. Some examples would be sugarcane and corn. C4 photosynthesis occurs both in the mesophyll and the bundle sheath cells of C4 plants. Initially, C4 plants are going to use an extra enzyme called PEP carboxylase, which stands for phosphoenol pyruvate carboxylase, to grab a hold of as much carbon dioxide as it can from those mesophyll cells, gathers it up, and will convert it into a 4-carbon compound called oxaloacetate. It will then be able to deliver that carbon fixed and that 4-carbon compound to the bundle sheath, where it can be released and then used in our traditional Calvin-Benson cycle to be able to produce that glucose. So we're going to be avoiding photorespiration by basically delivering the carbon dioxide directly to where it's going to be able to be used, so that the rubisco won't be able to have a chance to pick up any oxygen and avoid doing photosynthesis. Another alternative pathway is CAM photosynthesis, which stands for crassulacean acid metabolism. It gets its name from the family of plants, the Crassulaceae, which perform this form of photosynthesis, including cacti, jade, and aloe. So while C4 plants separate photosynthesis by location, CAM plants are going to be separating fixation by time. Part of what the process will take place at night, and part will happen during the day. The way CAM photosynthesis works is they're going to keep their stoma closed during the day so that they're not going to lose any water, and then they'll only open them at night. At night, the carbon dioxide is going to be fixed into a 4-carbon compound called malic acid. Then, during the day, when it's hot outside, they'll be able to keep their stoma closed, but then release their carbon dioxide so that it can then be used to perform our traditional photosynthesis. So by keeping our stoma open at night, we avoid our loss of water, and then capturing as much carbon dioxide as we can at night, we avoid photorespiration during the day. So each form of photosynthesis allows for the plant to be uniquely adapted to its environment. In warmer and drier climates, C4 and CAM photosynthesis allow for plants to avoid photorespiration and maintain high levels of photosynthesis.